my talk is not the title of the abstract. And I'm sorry I just sent in a scientific abstract. I don't think I'll have much time to talk about all the science that I presented in that. So the title instead is, the last thing I wanted to do was always the thing that I ended up doing next. And this harks back to what we've been hearing from just about everybody. Namely that science isn't really a career in the way that other professions are a career, but that we've all bumped around in our lives from place to place, and many of us without much planning, and I'll give you some examples of that. But I also wanted to say that about the talks we've heard so far, Ron yesterday said he wanted to inspire you. I don't know if you were inspired, but I really was inspired. I was also inspired by Vijay's talk, and I think this has been a fantastic meeting. And I'm particularly pleased that the really top people who are here are not the alpha baboons. I mean, the organizers must have selected the people who came for their really lovely interacting personalities and respect for science and respect for the community. I really appreciate that. So, how did I get into science? When I was about 15 or 16, I told my father that I wanted to be an interior decorator. He just came down on me like a ton of bricks and said, you know, why are you doing high school if you want to do such a silly job? And so I said, okay, I'll go back, finish my high school degree, and we'll see. In parallel, I was doing a lot of dance. I did a lot of ballet training, jazz dance, etc., and considered the idea of becoming a professional dancer, although I was a little bit too tall at that time. Dancers were tiny, so I was very worried about that. My feet were too big too. Nevertheless, I trained hard and started to look out for academies to start dancing. But then in the last year, I sort of didn't make very much progress in ballet. I didn't get much better at the school that I attended in my hometown. And so when the time came, I thought, oh well, okay, off to university then. And like Mike Eisen, I was very good at mathematics. I bet he's tons better than me, but I was the best at school in mathematics. I enjoyed mathematics very much. And I had a biology teacher who was really a wonderful teacher, sweet little old lady. I mean, she was tiny and sweet, and I think she'd spent some time in India. And so we were in a girls' school, and she taught us things like the best way of emptying your bowels, and she kept telling us that the way people crouch in India is much better than we sit on toilets in Europe. Unbelievably, in the 60s, that kind of thing was said at school. But so she was wonderful, and so you know, I went to. I decided to do math and biology. Actually, I want to briefly say what will come out of this talk. And I don't think there's going to be much of a take-home lesson, but I think what I'll try to say is that what I've done throughout my career and what's been good for me, but we've heard yesterday what's good for one person is not necessarily good for another, nor does, if I've had any success, does it have to be blamed on that. But what I'll try to emphasize is, what's been important for me is to interact with lots of people, with intelligent people, with wonderful people, with admirable people. I've had a lot of fun in my career, and I think it's important to have fun, and I've had very hard times, and I've worked through them, and I think it's important to work hard. So, interact, have fun, work hard. Okay, so I inscribed for a degree in maths and biology with the ultimate school, aim of becoming a high school teacher, and got my degrees in Bonn and Heidelberg, and started studying maths and biology. Like Mike, I discovered that maths at university is a completely different thing. At school, I never had to do my homework. So my best friend from school actually decided to go to Munich and study theater and choreography. And so I visited her a lot and started taking classes in ballet again with her there, with a teacher who was so fantastic that I made tons of progress in a very short time. And so when my friend changed to Cologne to attend the dance academy there, I went with her, and I was studying in Bonn, and I would go to my math lectures in the morning, because you could not miss a math lecture. If you missed a lecture, you were out. However, the biology lectures were so dull and boring that I decided to skip them all and go off to Cologne and take dance classes, professional dance classes instead. That went on for a while, and after a bit, and the teachers in Bonn at university, they didn't care. But the teachers at the academy in Cologne sort of called me into the director's office and said, what is this? What are you doing? Are you becoming a dancer, or are you studying biology and maths? And I said, well, and they said, you have to make up your mind. I said, and they said, are you good at biology and maths? I said, yes. And they said, well, then maybe that's what you should do. So they were quite clear that I wasn't cut out for dance. And so, so I went on and did my maths and biology. So, you know, I think this is typical of everything I've done in my life. You sort of bumble through, and eventually a decision happens, and it's not the decisions that you took before. I didn't take my studies in maths and biology terribly seriously. I spent most of my time having fun, doing lots of other things, continuing to dance. I danced in Heidelberg then. But the time came, actually there was one course that was really highly inspiring, and that was taught, a practical course that was taught by 2 immunologists who came once per year to Heidelberg to teach a practical course over 3 weeks, doing experiments where we really spent all, it cuts in and out, does it? Yeah. Spent all day in the lab until late at night, and then they went out and drank beer with us. It was the most wonderful thing. And in fact, one of them, Fritz Melcher, suggested that, you know, he asked me why I wanted to become a teacher, because he saw I was enjoying this work, and didn't I want to come and do a thesis in his institute. So I said, no, I'm going to become a teacher. I got my degree, and then I looked at what would happen next. And I saw myself going to school, teaching the kids in the morning, correcting their homework in the afternoon, preparing the class for next day. Next day to school in the morning, the same thing over again. Next week, the same thing over again. Next month, next year, for 40 years. And I just panicked. And I said, no way. So I didn't become what I trained to do, what I wanted to do. But I called up Fritz and said, look, you said I could come to your institute and do a thesis. Would that still be possible? And he made it possible. And so I went to the Basel Institute for Immunology, which was an amazing experience. And I list there my mentors. Fritz Melcher was my PhD supervisor. Jan Andersson was a colleague who actually came from Sweden, who was a fantastic colleague and really wonderful advisor. The Basel Institute for Immunology was really very special. It was founded by Niels Jerne, who later won a Nobel Prize. It was funded by Roche, and its structure was that there were 50 scientists. All those 50 scientists were PIs, and what they had was a lab and a technician and unlimited funds. So they could ask for any money they liked from Roche. There was a fantastic infrastructure. All mouse work was being done for them. All media were made. Fax facility, everything was done for you. Radioactive lab, everything was done for you. But the idea was that Niels would identify the best brains in the world, get them there, give them complete freedom. Usually these were postdocs. These were people right after their PhD. So you can imagine the atmosphere. The best people in the world with complete freedom. And there were 3 graduate students, and I was one of them. And now I came there with the idea, I'm sure you had a similar idea when you went into science. Science is where you think deeply and have a very good idea and think of an incredibly clever experiment, and then you go to the lab and do that experiment, and 3 or 4 days later you get a result, and it enlightens you, and you think again hard and come up with the next idea. Well, as you know, it's not at all like that. The trouble is, there were only 3 students there. And so, of course, I experienced that this was not so. But I saw that everybody around me was publishing wonderful papers all the time, giving fantastic talks. So I thought it was me. And it was really one of the toughest times in my life to recognize. And I had nobody else to talk to. You know, the other 2 students were somewhere else. They were also already fairly advanced. So that was really tough. I had a good time anyway. I stuck through it. I had long periods of serious hardship, but I stuck through it. And in fact, there were the good times. There was the first paper. I remember publishing this. Fritz encouraged me to publish, you know, my own results very early on. with a figure that I made with my own hands. Seeing that first figure you made in print, isn't that wonderful? So that was one thing. The other thing, and I learned many things from Fritz, the other one is that Fritz was, you know, he was really a great mentor. He gave me a lot of freedom and a fair amount of advice, among other things, by which men in the Institute not to get seduced. Again, completely impossible for a male mentor to tell a PhD student that now. He was right to tell me that. Anyway, so one principle he had was that every PhD student had to publish one paper on their own. One of the main findings from their PhD as a single author. And here it is. And that again was a big learning thing, so I had to write it by myself, I had to make the figures by myself. Of course, he advised me. And then I gave the manuscript to another scientist in the Institute to read it. Got it back after a week, full of red ink. Well, the first 3 pages were full of red ink, and then it stopped abruptly. And at the bottom there was a line by Stephen Fazekas-Sanko, who said, and he told me, said, "It is an outrage that you're expecting me to read this nonsense. I cannot handle anything of this quality. Never give me anything to read again, and don't dare to put my name in the acknowledgements." So it was a tough lesson, but of course he was right, and I went back and shared it with others of, you know, not of quite that caliber, and worked hard at it. And I've actually, we were discussing this afternoon, to this day, I give my papers to people, not only in the lab, but to outside people to read for advice. And it's always helpful, and I encourage everyone to do that. Okay, so during that time, by the way, of course we had no training for students. There was just no training for students. So I did set up my own journal club with the other students, with the other 2 students that were there. I also often traipsed across town to the Biocenter, where a lot of exciting science was going on at the time. Jeff Schatz was there, Walter Gehring, so some of the real big stars in cell biology and developmental biology at the time. And I learned a lot, and I met lots of people there as well, and I'll get back to that, why that's important. So at the end of my thesis, I was sort of, I was a bit worn down. And I actually, as the time came to decide what to do next, I got a bunch of catalogs and looked up ads for dance jobs, either on cruisers, or I had an offer from a musical hall in Berlin. But I also thought, you know, there is something in science, at least I'll try it, especially because the Basel Institute paid their students to make trips to any labs where they wanted to interview. And so I decided to try that. And how did I find out what I wanted to do? What I did was I actually sat down for about a week and took out every copy of Cell and Nature from the last year and started reading every single abstract and just triaged to find out what was definitely not interesting, sorted out a lot, and ended up with a few areas where I thought the topics were interesting, and then started looking where were the people who did the interesting work. And so I chose, because I knew one thing for sure, namely that I've been working with antibodies, I've working with tissue culture. It was so hard that I never want to do that again. No more monoclonal antibodies, no tissue culture. So the interesting things at the time were transcription, and so I went to interview with people like Wally Schaffner, Pierre Chambon. I also thought development was very exciting, so I went to see David Horowitz in London, and I wanted to work with Peter Lawrence in Cambridge. Peter didn't have any space in his lab, but he said there's another guy in the institute on his floor who's looking for a postdoc, and they found some interesting cell surface antigens, and I might be interested in working on them. They'd made monoclonals. When I heard the word monoclonals, I said, no way. But of course I wanted to go to Cambridge, so I said, yes, I'll interview with a guy, just as an opportunity to go to Cambridge. And I visited all those labs, and it's really important, I think, for postdocs to go and visit labs. I enjoyed many of them. I went to Cambridge to meet this person whom I hadn't selected, Mike Wilcox, and on whose project I didn't want to work. And you can guess You can guess where I ended up. Of course I ended up in Mike Wilcox's lab, on the one thing that I said I'd never work on again. But I just liked the atmosphere best, I did like the project, I loved the lab, and Cambridge was sunny for the whole 3 days I was there. I think the only time never happened again for the next 5 that I spent there. So that's what I did. I did my postdoc. I switched completely from immunology to development biology, and spent 5 years at the LMB in Cambridge. What does that computer have to do with that? Nothing directly, really. It's there to remind me that also in Cambridge, interaction with other fantastic scientists was one of the big deals and one of the greatest benefits I had from that time. And it's actually in Cambridge that I met Tim Hunt. And Tim Hunt was the first person I knew that had a Mac SE20. And when he got this Mac SE20, he invited a bunch of us into his apartment at Clare College, I believe, and we all sat around the table and drew doodles with the mouse of this fantastic machine. Do you remember that, Tim?
I do, yeah.
Yes.
Yeah.
So, again, I also...
We made up figures on it, I think.
We made yeah, we did all sorts of crazy things. I think
and finally, Baruch Benacerraf used one of those figures in his Nobel lecture or something.
Fantastic, yes. So that was Cambridge for you. Again, also there, going across town to seminars was a big deal. At the Department of Genetics, there was a weekly seminar where I saw, again, Tim show for the first time the gel that he brought back, basically still wet from the developing machine from Woods Hole when he discovered cyclin. Jim Smith came and showed us his first results of activin in the frog, and tons of other fantastic interactions. So these places are just wonderful, and one profits from them for the rest of one's life. So my work there was on these cell surface antigens that have been identified for using monoclonal antibodies. And what was interesting about them was that these cell surface antigens defined compartments in the insect body. And so we wanted to know what they were, what kinds of molecules they were. So I did a lot of biochemistry, ran lots of 2D gels and other things. We began to suspect that they were similar to integrants that were known from vertebrates, but we really needed to prove that. And in order to do that, we wanted to clone the genes, and in order to do that, we tried to get a protein sequence, which is not so easy, which was then not so easy from cell surface antigens that are not very abundant. And even if we got protein, it was very hard to sequence. So this is where it came in useful that I had a friend whom I'd met when I traipsed across town in Basel, Rudi Ebersold, who had at that point moved to Lee Hood's lab, where he was working on developing Western blots, blotting proteins onto glass fiber filters, which could then be used stuck directly into protein sequences. And I bumped into him somewhere. He just sort of begun to work it out. And he heard about my troubles about getting a peptide sequence from our cell surface receptors. He said, well, why don't we just try it? And so I, instead of blotting on a nylon membrane, as I've been used to do, or actually it was, what was that stuff that exploded so nicely? Exactly, nitrocellulose, which the last bits of one could always explode in the Bunsen burner and hope that one didn't set off a fire alarm. He sent me this fantastic glass fiber. I blotted my gel onto it, sent it to him, he sequenced it and came back with a peptide. It was wonderful, and here's the paper, in fact, in a journal. And that was the first time that was done, and it gave us a peptide sequence. However, I tried again and again, even with that peptide, I was unable to clone it. And it's particularly unfair, because as it turned out later, when somebody else cloned that gene, the first 10 amino acids actually have my name in it. It was meant to be my gene. Not a joke. You can look it up in the database. Okay, so that wonderful time too had to come to an end. Actually, what did I do for the community there? I don't know. I had a journal club there too. Again, there weren't many, but that was really basically Cynthia Kenyon and myself, we had a private journal club, which was very intense. We managed to read Ed Lewis's Nature paper, which is an achievement. Very few people have done that. I bet you have. But what we did for the community was arrange parties, and I think that's actually an important part for science. And I think the one most people will remember is the 25th anniversary party of the LMB, where the eggheads and alpha baboons had decided to have a fancy party in one of the colleges in town and invited Jim Watson and whoever else, and Watson gave a talk that was so unbearably awful. Well, I mean, everything he does is awful. Never mind, most people weren't invited. I don't know how I got a ticket, but most people of the LMB weren't invited. It was just for the eggheads. And so Peter Sawyer and I said, no, we won't have that, and we organized a huge party which got everybody involved. And I remember that, I mean, yeah, there's two. And so that's another thing one can do for the community. It doesn't have to be journal clubs, it doesn't have to be tutoring. Having fun, I think, is a good part of science. So I hadn't been to America as a postdoc, so I hadn't done my American stint, and I therefore really wanted to go to America after the UK. And I started applying for jobs, and I got a couple of interviews or so. But at the same time, a young graduate student from the lab of Jannie Nusslein-Vollhardt in Tübingen, Ruth Lehmann, had come to the lab to learn molecular biology, to clone one of the genes that she'd identified during her thesis. She later then took that to the Whitehead Institute, and where she did all her famous work on nanos, and that's what she cloned in Cambridge, and on her developmental biology work. So she then heard that there was a job available in Tübingen and said, Maria, you must interview for that job. I said, I do not want to go back to Germany, no way. Another friend of mine, Vivian Siegel, who'd come from Peter Walter's lab and later became the editor of Cell, was in Tübingen doing a postdoc with Jannie, and she did the same thing. She said, Maria, here's this job, you must come to Tübingen. So I said, okay, I'll interview. And I looked at the job. Of course, it was a nice package. I didn't want to go there, so I went to interviews in America. But then eventually they pulled too strongly. It was too good an environment. It was too good a chancellor. So I went back to Germany. The Max Planck Society were actually very nice. I told them that I had really wanted to go to America for a while at least. And what they allowed me to do is to use the first 3 months of my time there to do a micro-sabbatical at UCSF. And I went to the lab of Pat O'Farrell to do some work. And actually my 3 months and the year after I went for another 2 months at UCSF really, I would say, fit perfectly what Mike yesterday quoted Sidney Brenner as saying. It's best to work on something. Best time to work on something is before anybody else is interested or after everyone has lost interest. And I did both. The thing is, there's a caveat to that. If you do that, you actually have to accept that most of your peers aren't interested in talking to you, and the journals aren't interested in publishing your stuff. So what did I do? These antigens that I'd been looking at in Cambridge turned out to be integrants and therefore cell adhesion molecules. And they had a very interesting complementary pattern of expression in the early embryo, which to me said that they would have to be involved in gastrulation, in making, in creating the different cell sheets in the embryo, as they had been sort of shown in vertebrates. I did all the genetics, and it turned out they didn't. So I was wrong about that, and I published a not very nice paper, but they were not interested in gastrulation. So I became interested in what else might do gastrulation then. And I started talking to Pat about this, and he said, well, something must be different in the cells that go in and the cells that stay outside. It's very early in development, so the proteins probably all exist. They're just modified in different ways. What does one do to look at differently modified proteins? Well, one does 2D gels and looks for phosphorylation patterns. So Pat O'Farrell, does anybody know what Pat O'Farrell was first famous for? He is Mr. 2D gels. He invented 2D gels. And Pat still had his old apparatuses sitting somewhere in some shelf. And so he got them out and said, let's do this. Why don't you just look at embryos that are, and I could do the genetics. I had embryos that behaved just in one way and others that did, and do differential gels. So when I started talking about using 2D gels to do this kind of thing, people's eyes just glazed over. You know, you would go to these wonderful seminars that Ron mentioned that everybody went to, and you hung around. There was always cookies in the back afterwards, and so you'd hang around, and people would talk about it, and as soon as I said this, they would sort of turn away and talk to somebody else. Which I think is a bad thing, because there is a mainstreaming of science that can be very bad in big institutions. However, it meant that I also talked to other people, and I listened to what they did, and I ended up talking to people who were, first of all, there was John Sadat's lab, who was building new microscopes that were fantastically exciting, and there were also biochemists who'd been able to make fluorescent molecules that could be watched in vivo. And what I ended up doing was to actually observe gastrulation, not look at it biochemically, but look at it down the microscope. Now, you must remember, this was way before GFP. So, of course, I had observed embryos in the microscope with transmitted light, all the sophisticated slanting illumination, but you couldn't do fluorescence microscopy. However, these clever people had actually made fluorescent actin, which doesn't work very well, fluorescent tubulins, and fluorescent histones, which you could inject into the early embryo, and then you could observe all these things. And you could also inject fluorescent dextran in the space that surrounds the embryo, which would then be drawn between the cells. So we were able to make a two-color, so we used green dextran and red histones. We were able to make a two-color movie over time with John Sadat's new confocal microscope. Really, I didn't think about it as pioneering. I only realized much later that, in fact, we'd made the first 4D movie of Drosophila. And John Sadat's clever people were able, so what you see here approximately, you see in one row the cell outlines and then the cell nuclei, and we could look at them at serial sections, and they were able to reconstruct the cell shapes, the cell outlines, and the nuclear positions in them. And in fact, we then used the nuclei as proxy for cell shapes, and this allowed us to figure out what the embryonic epithelium, which you see at the top, how its cells begin to move inward when when gastrulation begins. I'm way behind. Okay. We did think this was a fantastic piece of work, and so we sent it to Science, got it back with 2 raving reviews, and one sort of lukewarm review, probably from some idiot who'd never heard about gastrulation, wasn't interested in development, and had only done microscopes or so, was probably a microscope expert, who thought the story was boring. Well, we submitted. Elsewhere it got in. A couple of months later, I meet my dear old friend, Louis Wolpert. Now, I don't know if who of you know Louis Wolpert, one of the most famous development biologists, big book on development, and the statement that it's not birth or marriage that's most important in your life, but gastrulation. And he said to me, "Oh, Mariah," he always called me Mariah, "I recently had to review a paper of yours. Now, you do beautiful work, but that, this microscopy, that really was below your standard. So I'm afraid I had to reject it. It wouldn't have done you any good to have it in such a high-profile journal anyway. It would have damaged your reputation." Well, tough luck. The other thing that happened in San Francisco is that I met my husband. No, I didn't meet my husband. I married my husband. I met my husband earlier in Cambridge, but we separated after my 5 years. But after a year, we decided separation wasn't good, so we got married. This is a year after the earthquake in San Francisco. They had not yet repaired the city hall. Okay, so I then went to Tübingen. I have to race through this, where I started, which was fantastic. This was the time when all the developmental biology, the pattern from molecular biology of pattern formation was being done there. The bicoid gradient, the dorsoventral gradient, toll signaling, cactus, you name it. And the group of people there were just fantastic, and it was really 24/7 hanging out together talking science. This is where I started working on gastrulation. Then I proposed a project on the integrins that I've been working on. I ditched them. I started working on gastrulation. So again, I did something completely different from what we thought. I'd we figured out that cell shape changes in the cells that I was interested in brought those cells inside. I was extremely lucky to have a technician who was able to make these beautiful, she was a fantastic histologist. I was lucky again. I had a colleague, Siegfried Roth, who made this beautiful antibody that stained the cells of interest. And so we put together this whole story. We really figured out how the mesodermal cell layer is made in the Drosophila embryo. So I had a very productive time there, and I think I did some of my best work there. I was actually also productive in other ways. So I had 2 babies during the time there, which was not that easy because my husband lived in Cambridge and I lived in Tübingen. And making babies is easy, but it's not easy if you only see each other on weekends and when the day you're supposed to be seeing each other is on Wednesday. Nevertheless, we managed to make 2 nice babies, and we took turns looking after them. So, it was 2 weeks apparent, and then I'd go to Cambridge and then leave the kids there, go back to Tübingen, be a postdoc for 2 weeks, and then Jonathan would come back. We'd spend some time together, and then he'd go back to Cambridge to be a PI. Well, that couldn't last, so we both looked at various job offers in London. I wanted to go back to England. I loved England. Cambridge is great. London is great. We had some job offers, but somehow, eventually, and I wanted to, of course, go to a research institute. My husband was asked to apply for a professorship in Cologne. I said, professor, last thing I want. My father was a professor. University, not for me. You know my refrain. We ended up in Cologne as professors. We moved together, and this was actually an easy time. When we moved to Cologne together, both had to start a lab, had 2 babies, no childcare. It was the most nightmarish time in my life. That was tough. That was the only really tough time I had in my life. Also, I'd been moving every 5 years. I swore I'd not move again for at least another 10 years. Well, I took 18 years before I moved again. I did a bunch of things there. I founded the first international graduate school in Germany at the Institute of Cologne Genetics. I didn't like the way that graduate students were being supervised, selected, mentored, so I started an international graduate school, which eventually led to big changes in graduate training overall in Cologne. One of the first students in that graduate school was Sam Matthews, who's now a PI here in India. Other lab members from the Cologne time also become PIs. Maitri Narasimha at TIFR and Renjit, who was here until yesterday. He's just started his lab at ISER in Bhubaneswar. And I was hoping to go through the science a bit, but I think I'll have to skip that. Oh, another thing I did in Tübingen, in Cologne, is to use the freedom one has as a university professor, is just to go off on tangents and do completely new stuff. So during a midlife, actually I did various other things, and during a real scientific midlife crisis, I started working on fish immunity. This image shows that baby zebrafish can kill bacteria, but not if you compromise their immune signaling. Now, every scientist says one thing they don't want to do is administration. And so why was I so crazy as to take on an administrative job and become director of EMBO 5 years ago? Well, I knew the organization. I'd been on their committees. It's a fantastic organization. It's the 1,600 leading scientists in Europe. It's an office with staff that is dedicated and wonderful. They have fantastic programs. I'll tell you about them tomorrow. And again, I was a little bit pulled by friends who were at that time in charge at EMBO, including Jannie Musslein-Volhard and once again Tim Hunt. And I have to say, I don't regret it one bit. It's also not so much an administrative job, it's really a strategic job. Plus, they threw in a lab at EMBL to sweeten the pill, and that's just been fantastic because the possibilities there. So if I had time, I would tell you the science I've done there, but I think I'll just stop there, and maybe I'll just mention it's all models films. Yeah, sorry. and just thank people in the lab. So, as we all know, lab work is teamwork. So this is the lab last year at EMBL when we're about to go on a lab outing. That's my car, a little Ford car. What do we do when we go on a outing? Well, we all pull together, as always, get in the car and get going. And the only thing that's wrong on this on this image is that I don't have to drag the lab. They are there, they inspire me, and they push me, as does everyone here. So, thank you.